You want it on? Actually, I'm having sore throat, so that's why I'm.
That's okay. That's okay.
No, no, no. I have water here. Even I feel hot. Once I start speaking, it will become very hot. You all saw your test papers. Some of you have really got confused with epimers and that topic, right? Some of you have got really confused. You have answered wrong answers for epimers and that question where you have first and foremost, you have the answer sheet with you. Those of you who have, you can just take out the very simple 5 questions I asked because I had not met the other batch students. So I had asked for just the basic questions and still I was surprised that some of you really gave wrong answers in those. I so clearly mentioned as to the position of H and OH group. Okay, for the 5th carbon atom in glucose will only say whether it is a D or L isomer and not epimer. Isn't it? Epimer is for the 2nd or the 4th carbon atom. Right? 5th carbon atom is farthest from the carbonyl carbon and that will determine whether it is a D isomer or L isomer. And again, it is not dextro or levo. Alright, because dextro and levo is the optical property. This is the stereo orientation in nature. So there is 2 different. Dextro and levo is different from D and L isomer. Alright, then what was the other question? The answer is pectin. Alright, for the non-monosaccharide is pectin. What was the other question there?
Bipolymers such as hemicellulose, which is used in bioengineering. Structural model is important in bioengineering. Arabinose.
What were you telling me? Arabinose is the answer, right? Because there the hint is it's an L isomer. L isomeric forms I've already mentioned, right? L rhamnose, L fucose, L arabinose. In monosaccharides, it's only arabinose which occurs in the L isomeric form. Okay, then what other?
The monosaccharide that is optically inactive.
Yeah, tell me which is that? Why?
It is a ketone.
Yeah, the 2nd carbon atom there is the ketone. The 1st carbon atom? CH2OH. 2nd carbon atom? C double bond O. 3rd carbon atom? CH2OH. Okay, because all the 3 are not chiral carbons, so that is why dihydroxyacetone is the only monosaccharide which is optically inactive. Alright, then what was the another question?
6-membered cyclic structure of monosaccharide.
It's a very simple question, isn't it? 6-membered ring structure is pyranose structure. So there also there were a few mistakes.
Ma'am, those have chiral center, chiral carbon. They are only optically active.
Exactly. Any molecule which has got even one chiral center will exhibit optical activity because they will have a rotation in their functional groups.
Yeah.
Alpha beta has also optical activity also. They are also a kind of isomers, stereo isomers. Alright. So you were asking me about the peroxidants. Pro-oxidants. So I have an answer here. Pro-oxidants, well, I'm talking to you about bioenergetics. There I'll be telling you about the so-called free radicals which will be released in the biological systems. Alright, so there if you see, the free radicals are actually acted upon by antioxidants. Alright, so free radicals, particularly the reactive oxygen species. These reactive oxygen species are the peroxidants. Okay, you have superoxides, you have hydroxide ions. So about different free radicals when I'm talking hydrogen peroxide. So these are all the free radicals. They are the reactive oxygen species. Alright, and these will be acted upon by the antioxidants in nature. So because free radicals have got even positive roles and also negative roles in the body. When they are having negative roles, the antioxidants will inhibit their effect. Alright, so those are peroxidants. But this you are asking with respect to ascorbic acid. The question the other day I asked you, when you leave food stale, the vitamin content becomes inactive vitamin. So what is the reaction happening there? Oxidation. What happens to oxidation of ascorbic acid? What is the compound it forms?
Alcohols.
Alcohols? No.
Aldehydes.
Aldehydes? No. It forms dehydroascorbic acid. Alright. When oxidation of this is a reversible oxidation. That is the first step in which ascorbic acid gets converted to dehydroascorbic acid. It is reversible. It can become ascorbic acid once again. Alright. Further, if the food is getting stale, it's still remaining stale, again it will get hydrolysis. It will undergo hydrolysis. And then it will form diketogulonic acid. Alright. So that is the vitamin inactive. The diketogulonic acid is the vitamin inactive form of ascorbic acid. That happens when the food becomes stale. Alright. Arabinose. Yes, it is arabinose. I just mentioned, I just discussed the paper, Praveen. These are very simple questions for your batch, I suppose. Right? Because the same thing I have repeated several times, particularly for your batch. So even then, there are a few mistakes. If you still have doubts with respect to epimer, anomer, and enantiomers, you please go through once again 2 or 3 times, try to understand, and if you still don't get it, then come back to me. Alright? So the other, and then I was asking you about lactose intolerance, right? We'll just have a brief discussion on the doubts. What are the tests conducted for lactose intolerance? One of you asked me the other day, right? So did you try to find out? I said we'll collaborate some information. No? No? Anybody? Yeah, tell me, Lavanjika, what are the 3 tests? Anybody here? What are the tests that are performed in order to determine whether a person is lactose intolerant or not? Digestion of starch. Elisa, Arjun, you have told me that previously. Right, that's a very elaborate test. How do you immediately come? No issue. Come fast. Tell me. Lactose intolerance test. Exactly, you're correct. Can you elaborate? Lavanjika, have you identified the answer for this? Momita? Tell me. What is lactose intolerance test, hydrogen breath test, and stool acidity test or stool sample test are the 3 basic immediate tests. As soon as you have a suspicion that you are having lactose intolerance, the first thing the doctor will do is to perform these 3 tests before going into more elaborate ELISA test, etc. So, meanwhile, as Lavanjika is elaborate, Lavanjika. Blood test, yeah, Arjun, that's the blood test is the lactose intolerance test. Fine. So by the time Lavanjika is elaborating, I want one of you to come out with the answer how starch is getting digested. Amylase enzyme. Step by step, tell me what happens as soon as you have taken potato into your food today. Early morning tiffin, you have potato. So tell me what has happened to that potato. As soon as you take, the salivary amylase will come. First you chew the food, you break down from complex to a little simpler. Then what happens? Alright. Okay.
It will break it into maltose.
Okay.
Then simple
Sugars in the mouth itself.
Then it will reach stomach.
Okay.
Stomach there won't be any action for the carbohydrate.
Okay.
Then again the small intestine, the pancreatic amylase will convert carbohydrate.
Then pancreatic amylase, then finally intestine. Anybody else? Elaborate on what she said. Here online students also. Hydrogen level, see they are giving the answers. Okay, for lactose intolerance test is the test in which the blood sample is collected and the level of glucose is identified. Exactly how Lavanjika has told that the blood level, the glucose level will increase only if lactose is broken down, isn't it? If lactose is not broken down into the normal procedure and if it is undergoing fermentation, then your glucose level is not going to increase. So the normal glucose level content in the blood is how much? How much? 32 mg per ml is the normal quantity of glucose in our blood. Alright, so this quantity increases only when your food is getting converted into the monosaccharide. When that does not happen, okay, when that does not happen, then lactose, the glucose level does not raise. That means the lactose has got fermented. That is the lactose intolerance blood test. Then the other test is the hydrogen test. Alright.
So ma'am, here the glucose level decrease or increase?
It does not decrease, neither it increases. The glucose level is normal. Or it is remaining, it's not increasing even after you have consumed the lactose rich food. Alright. Now the same thing for stool sample test. It contains lactic acid if lactose is not digested. Exactly what she has told, that for particularly for children. Alright, infants, you cannot perform this lactose intolerance test to a great level. So what they do is they collect samples of the stool of these children where their lactose is not digested well. So undigested lactose is fermented and acid content in the stool increases. Lactic acid and several other acid content will increase in the stool. So that is one way of detecting the lactose intolerance. And then you have the hydrogen breath test. Okay. Here, hydrogen breath test is where the level of hydrogen is checked. Exactly. See, first, you are allowed to normally breathe into a balloon-like bag. Alright. And then the hydrogen content is identified. Then after you consume some amount of lactose, then again you are allowed to go through this. You are allowed to breathe into that bag. Alright, so if you compare, normally when we breathe, some small quantities of hydrogen concentration is up and down in our breathing system. Traces of hydrogen you can always find in our breath. But when the food is not able to break down the body is not able to break down its food correctly, then the hydrogen content in our breath will increase. And this level of hydrogen is what is detected. Okay, so if lactose is not being digested properly, then the hydrogen level in our breath will increase. So the percentage of hydrogen will be compared from normal to this. Alright, so for lactose intolerant person, you will see that level of hydrogen has increased in his breath. Alright, so that is about lactose intolerance test. Hydrogen level increases because of the intermediate, because fermentation. When lactose gets fermented, alcohol is being produced. Alright, when alcohol is being produced, particularly in the intestine, then several gases will be released. Right? You see, when a person does not digest food, that person will release lot of gases. Right? So that is a suggestion that the food has not been digested properly. So that way the level of hydrogen will increase in the breath of that person. Alright. So those were the three. These are just the additional informations. Okay. And apart from that, I was talking to you about digestion of starch. Right. Steps in the digestion of starch. There we see that the starch, when it undergoes digestion, first thing, as soon as you take starch, you are masticating. That means you are chewing the food and your teeth is helping to break it down into simpler molecules. But still it is a polysaccharide. Alright. Then the first set of enzymes which acts on the starch is the amylase enzyme, which is present in the saliva. So what this amylase enzyme does is that it will hydrolyze the glycosidic bond. The alpha-1,4 glycosidic bonds, what are present in the amylase, what are those? Amylopectin,
amylose and amylopectin.
These two are the subunits of starch, right? So on the alpha-1,4 linkage, the amylase enzyme will act on. Alright, and there some of the glycosidic bonds will be broken down. So hydrolysis means the water molecules will come and cap around the glycosidic bond. Alright, so that will allow the reactive species, the hydrogen and hydroxyl group, to be available. It will cap around the glycosidic bond. Okay, then the food goes down your esophagus and it comes to your stomach. In the stomach, it's not digested, it is not broken down, but it is acid treated. So what this acid does is that it will inactivate the amylase enzyme. Alright. So long chains of polysaccharide, what you have taken inside 1000 glucose residues linked to one another. Once it reaches the stomach, it would have become around 8 glucose residues linked to one another. Okay. As less as 8 or 10 glucose residues linked will be in the stomach. Then over there you have the pancreatic amylase. Again, these are amylase. They work very similar to the salivary amylase. Alright. So these pancreatic amylase will again break down these into dextrin and maltose, maltotriose. Maltotriose means 3 saccharides. From 8 glucose residues, it is coming down to 3 glucose residues. Then you have dextrin. Dextrins are talked about. What are they? They have branchings. Right? Extensive branching will be there. So wherever you have this alpha-1,6 branching in the amylopectin, those will be separated out from small stretches of oligosaccharides and the branchings. Okay, so you will get dextrin, you will get maltose in the blood sugar. Alright, so that is the action of amylase. Then further in the intestine. In the intestine you have enzymes like glycosidase enzymes, dextrinase enzymes. So these enzymes will break these trisaccharides into individual glucose residues. And these individual glucose residues will enter our bloodstream. Alright, so this is how starch is broken down, not by just one step, but series of steps in our body. And that's how a complex polysaccharide is broken down. Alright.
And for this, which book can we refer
For all, these are all general informations. Anywhere you go and you try to find out about digestion, you will get information. Alright.
Ma'am, you said several gases are increased in the stomach. So, ma'am, when food is digested already, the function of gastric juices.
You're talking about lactose? You've gone to lactose or?
Yes, ma'am. Lactose.
Okay, tell me.
So, gastric juice, presence of gastric juice break down the food.
Yes.
Or else food hydrolysis.
Yes.
And how about the gastric patients?
They are all gastric patients. Again, that is a gastrointestinal disorder, isn't it? Even though you have correct actions, all these are coordinated. Several coordinated enzymes, several coordinated biomolecules, everything will have to come into play. Where there is a defect in any one of the enzymes or any of these biomolecules, there you will see this kind of an effect. Right? Gastrointestinal disorder is only the gastric problem what you see in people. Right? So that is their increase in the hydrogen content. This is not only for lactose. Any kind of indigestion, when the breakdown of food does not happen in the normal method, immediately there will be an increase in the hydrogen level. So that is to test if the person is digesting the food properly or not. Alright.
Then how can we refer exactly that is happening due to lactose?
Because before that, first the test starts with fasting, where your hydrogen level will be detected. Then you are consuming lactose, then the level is detected. Then you will get the correct level.
Okay, then other food also will check and if
fasting means there is no other food before that.
No. Like if he is particular to lactose intolerance or he has got digestive disorders.
For any other food, that he will be able to himself tell you, right? When only I am taking milk product, I am finding some kind of. So then that person will go to the doctor and doctor will suspect that he must be having this lactose intolerance, right? Diabetes is completely Shivpriya, these are all additional informations. I'll start off with the class and I'll get back to you on this. Alright, you go to my previous recordings because today is your first class. You go to my previous recordings, you listen to those and you get back to me. I'll explain to you. So what were you asking, Jaskirat, about diabetes? Diabetes is completely a different concept. Alright, now if I am explaining on that, it will take more time. Just try to understand that the glucose level in the blood increases. That is the condition of diabetes, right? So there what happens is that the insulin, action of insulin comes, the pancreas, those will be very, completely a different topic. Do you want me to deal? I'll complete polysaccharides and come back to that. Okay, yes, a person who's diabetic is not supposed to consume starch because the number of glucose residues will become more in his blood. And they cannot break down, they cannot further digest this glucose. Alright, because of the deficiency of production of insulin in the pancreas. Right? So here I suppose the last class we were just talking about the various reactions of monosaccharides. Here today you have to remember certain names what we have previously talked about, particularly the amino sugars, N-acetyl glucosamine and N-acetyl muramic acid. So if you have forgotten those, alright, the change in the hydroxyl group at the second carbon atom will give the amino sugar. Alright, so some of them are acetylated in nature, like the N-acetyl glucosamine and N-acetyl muramic acid. These two are we are going to talk about them today extensively. So I'm just making you understand how they look. Alright, muramic acid, that is nothing but glucose residue at the second carbon atom has got replaced by an amino group, and at the third carbon atom it has been added by a lactic acid. Alright, and that amino group which is replaced in the second carbon atom is acetylated. So that becomes N-acetyl muramic acid, and N-acetyl glucosamine I have talked about. Then we saw oxidation. Oxidation gave rise to gluconic acid and also certain uronic acids like glucuronic acid. So isomer of glucuronic acid is iduronic acid. Alright, so even those will be seen in the structural polymers when we are going to study them further. Then we saw the reduction of monosaccharides gave rise to alditols. We saw several examples of these. Then we understood what is glycosidic bond with respect to disaccharides, right? And we saw several examples of disaccharides where we saw lactose and sucrose. We saw that in lactose the anomeric carbon atom is freely available, and that is why it is the reducing sugar, and in sucrose both the anomeric carbon atoms are involved in the glycosidic bond, and that's why they are the non-reducing sugars, right? So polysaccharides I started for you all, and I think we finished with the homopolysaccharides, right? So polysaccharides, homopolysaccharides, they contain only one monomeric unit. So there we saw a few examples of storage polysaccharides. Starch, which contains amylose and amylopectin, both linked together as a double helix, they are present. And then we understood the digestion of starch. Another storage sugar we saw in animal tissues, those are glycogen, and how glycogen is different from starch is that they are similar to amylopectin, except that the branching is more extensive, right? Then we saw what are dextrins. Some of the structural homopolysaccharides which we studied the other day, cellulose, right, where it is the beta-1,4 linkage. It differs from the structure of starch and glycogen in that it is the beta-1,4 linkage between the adjacent glucose residues. Right, then we saw chitin, which is the...
We didn't do that.
Oh, you didn't study the...
You gave them some examples of starch, glycogen and all.
Alright, the other day
It was just the difference of
okay, I think here only the system went off, right? The other day? Your class only?
No.
Starch and glycogen have completed in your class? No? Okay, we'll get back to it.
Just the difference we were done with. Oh, I'm not sure.
So I have not dealt with starch yet in your class, alright. Okay. No, why? I'll be dealing with it, don't worry. See, first of all, don't get tense because actually I'm going too much in detail for you all. Right? You're all master's level, so talking about starch, cellulose, glucose and all is too much preliminary level because we are just getting to know each other. I'm very slow in these topics. Alright, so I see how much you all already know. Starch, as I already talked about the digestion of starch, it's more rich in the storage forms in plants, right? Like for example, tubers, potato are very rich in starch content, right? And these occur as granules or clusters inside the cell. Alright, they are heavily hydrated in nature. That means they have several hydroxyl groups which are exposed in their structure, because of which they can easily bond with the water molecules. So that's why they are heavily hydrated. Alright, and starch will reduce the intracellular osmotic pressure. So if you have individual glucose molecules in the cell as the storage unit, then the osmotic pressure will increase and the cell will lyse. Alright, whereas starch, because of the presence of linkage, the molarity decreases and it will help in combating the osmotic pressure. That is why starch is an storage component. Alright, mainly because it can reduce the intracellular osmotic pressure. Alright. So how?
Please explain the concept behind.
See, glucose has got around 0.4 molar concentration, alright. Whereas for starch it is 0.01 micromolar is the concentration of it. Alright. So that way, if you see, glucose will has more concentration, and that is the solute concentration is more. Because of which the cell will lyse if you have individual glucose units. Whereas if it is present as starch molecule, then more of storage is possible. Alright, more compactness is possible, because of which the osmotic lysis of the cell will not take place. Alright. Are you all with me, online students? Alright. So let's see how this starch looks like structurally. Right? Starch consists of amylose and amylopectin. So what is amylose? Amylose is nothing but the unbranched chain of glucose residues. How are they linked to one another? By alpha-1,4 linkage. How is it different from amylopectin? In that amylopectin, you will see branching at the alpha-1,6 linkages, and that branching occurs for between every 24 to 30 residues. Right. So starch has two types of glucose polymer. One is amylose and the amylopectin. Now this amylose and amylopectin will interact with one another and form starch. Alright. So if you see the structure here, it's the amylose and amylopectin will form a double helix structure. Alright. Then amylopectin will also form double helix structure within itself. So this is how the compactness of starch is brought about, because of the interaction between the amylose and the amylopectin polymers which are present inside the starch. Alright, so how they are present? Like a V form. The amylose and amylopectin are interacting with one another such that it has got a reducing end and the non-reducing end. Alright, so particularly reducing sugars or non-reducing sugars we confine to disaccharides or monosaccharides, alright, but we do not come to polysaccharides to distinguish whether it's a reducing or non-reducing sugar, because in nature they are not present as individual glucose residues as an open chain structure, alright. They have severe conformations in nature which will help them carry out their biological role. So because of that, you cannot specifically distinguish them as the reducing sugar or non-reducing sugars.
Ma'am, branching 1,6 linkage at every 24.
Between every 24 to 30 residues of unbranched glucose, you will find a branching.
Full branch 24, then 30.
24 glucose residues, then you will see a small branching. Again, around 25 to 26, you will see a branching. Between 24 to 30, it varies from one starch to another. Okay, and digestion of starch, I've already talked about, right? In the beginning of class, I talked about how the complex starch goes down to become individual glucose monomeric units in the blood level. Then glycogen, right? So how starch is the storage polymer in plants, glycogen is the storage polymer in animals. And how is it homologous to starch is that they also are present as intracellular clusters or granules, and they are also heavily hydrated in nature. How are they in structure? They are very similar to amylopectin. The only difference is that they are extensively branched. So as I said, amylopectin has got what kind of linkage? Alpha-1,4 linkage. What is the branching? Alpha-1,6 branching. Okay, linkage is alpha-1,4. Branching is alpha-1,6. So use, as I said, between 24 to 30 glucose residues, those branchings are there. That is in the case of amylopectin. In the case of glycogen, the branchings are more frequent. Alright, that's the difference.
Linkage is like this, branching is from down.
Branching means between the first and the sixth carbon. Alright. It will form two layers like this. Okay, this is the branching. It can be between 1,3, 1,4 also, like in the case of dextrins. Alright, but here specifically it is alpha 1,6. And alpha 1,4 means it is adjacent linkage. So, how the branching occurs is between every 8 to 10 residues. 8 to 12 residues, you see the branching. It is the reducing end which is the extending end, and non-reducing end of the glycogen is the non-extending end where there is no free anomeric carbon atom. At the reducing end you have the anomeric carbon atom to which the glucose residues goes on adding itself. Alright. So where is the maximum presence of glycogen? It is present in the reducing end. More of it is see, let me explain the structure once again. So it is nothing but same as amylopectin structure where the linkage between the individual glucose residues is alpha 1,4 linkage and the branching is at the alpha 1,6 linkage. Now for every 10 such residues, glucose residues, you will see one branching. 8 to 10 or 8 to 12 glucose residues, you will see such branching. So because of that, it is extensively branched, and because of extensive branching, it is more compact. Clear? So where do you see? It is found in liver. One of you asked me a question the other day, right? From the study portal as to glycogen is present in liver, kidney, not your batch. So glycogen is also synthesized in the stomach and brain also. Okay, you can see glycogenesis in the stomach and brain, whereas traces of it is found in kidneys also, right? So the main constituent organ in the body where you see glycogen is the liver, alright? Also in the skeletal muscles you see glycogen. Another type of polysaccharide, what we need to know is the dextrin, because we have seen some homopolysaccharides in plants, we have seen some of them in animals. In bacteria and yeast also there are certain homopolysaccharides. Some example is dextrin. Alright. So dextrin is also glucose residues. Alright. The polysaccharide, homopolysaccharide of glucose residues, but the only difference being that it is extensively branched. Alright. So you can have a branching between alpha 1,6, alpha 1,4, 1,3, 1,2. Any kind of a branching can be observed in the case of dextrin. Alright, so where you find dextrins more is the presence of it. You can see them in the bacteria which is growing in the dental plaques. Where else do you see dextrins? The use of dextrins, synthetic dextrins, right? Where Sephardex, size exclusion chromatography. What is that? Size exclusion chromatography. What is the basic principle? Based on size. Are you sure? Based on the molecular size. Alright. So the dextrin molecules will have different porosity because of several cross-linking. This cross-linking is because of so much of branching. Extensive branching gives rise to extensive cross-linking. And extensive cross-linking will give different porosity. So because of this porosity, it is being used as a Sephardex in the case of size exclusion chromatography, in which you can separate the molecules from larger molecules and smaller molecules can be separated from one another. Alright, so that is the
Explain again, ma'am. Extensively branching gives
cross-linking. More the branching, more cross-linking will be seen. So because of that, porosity is there. Everywhere you can see some pores that will be present through which the molecules will pass. Alright. Then?
These pores because of the branches.
Yeah, more branching, more cross-linking, and that's how pores are also more. Alright, so that is the principle for which it is being used here. Alright, because through the pores the molecules will pass. Through the individual pores the molecules will pass and come out. Smaller molecules will elute faster. They will go through the pores and they'll come out faster. The larger molecules will take more time to elute out, isn't it? Right. So these were the storage polysaccharides, right? So now let's see some of the homopolysaccharides, the structural homopolysaccharides. Cellulose. Alright? Cellulose, where do you see them? Mainly in the plants, right? So the end of photosynthesis, you get starch or cellulose. So what is their role in plants? In the cell of plants. Structural. They are the structural homopolymer. That means that you have a tree, right? In the tree you see the trunk bears how many leaves on them, how much of branches on them. Look at the weight on top of its head, right? Flowers, fruits, the leaves, it has got lot of weight over it. So it has to have the ability to bear this load, right? Not only that, when a wind blows, a tree cannot get washed away, right, like that, right? So neither a small waterfall, rain or something cannot gush out the tree like that. So it is rooted, that is one reason. Another reason is the presence of cellulose. Alright, so this cellulose will confer rigidity to the trees. Okay, individual cells will get rigidity and several such individual cells together will confer rigidity to the entire organism. Right, so how it does this? It is very fibrous, it is tough and water insoluble. Alright, so as I already said, it has got load bearing capability and it helps in maintaining the osmotic pressure, the shape and rigidity and what makes it so rigid? Just the presence of glucose residues. Alright, it is just the glucose residues. But how these glucose residues are present? Not 1 or 2 glucose residues, but 10,000 to 15,000 such glucose residues are linked to one another. And how the linkage is, it is different in that it is a beta-1,4 glycosidic bond. Alright, so can we digest cellulose? No. Why? We lack the cellulase enzymes, right? So then why do so we cannot eat grass, but still we do eat some green leaves, right? We eat spinach, we eat lettuce. How? If we cannot digest cellulose, we cannot eat grass. How can we eat spinach or lettuce? Roughage, but it has cellulose, no?
Yeah, those which are not digested become like roughage.
So that means spinach is not digestible. Then why do they say eat lot of spinach?
No, few parts are, that is not only solely consisted of cellulose. There are other.
You're near the answer. You're almost the answer. Can you elaborate a little clearly? Somebody here, online students? Tell me. You were just answering, Ashmi.
And the other things are absorbed except for the cellulose, which acts as a
You're correct. Putting all your answers together, okay, cellulose is present even in the case of spinach. It is also present in the case of lettuce or any kind of leaf what we consume. Even the raw leaves sometimes we consume has got cellulose. But that cellulose portion of that leaf is not what we digest. That will only, it is very fibrous, right? I already mentioned cellulose is very fibrous. That will add to the fiber and roughage of our food. Alright, but why still they say eat lot of spinach is because apart from cellulose, sugar, it has lots of vitamins and minerals in it, right? So because of that, we have certain oxidase enzymes which can easily digest these vitamins and minerals. So because of that extensive presence of these vitamins, they ask us to consume spinach very frequently. But the cellulose part is only the fibrous and roughage to the food. Alright, so that's why generally you must have heard people telling do not consume spinach in the night times, right? So where your digestion level is low, so at that time they generally suggest do not consume them during the night. So this suggests that herbivorous foods are more difficult to consume, right, than the digestion of carnivorous food. Herbivorous food is more difficult to digest in this aspect. If you look at, like for example, cattle, the cows, how they eat the food? They go on, they chew for such a long time, isn't it? Apart from that, if you see their stomach, it is multi-chambered. Alright, so it has been specially designed in order to eat the grass. Right? So that much of complexity is there in this polymer. So who contains cellulose in the cattle? Is it the cattle itself which has the cellulase enzymes? Bacteria. There are some symbiotic bacteria in the gut of these animals which will digest these cellulose. Alright, the same way if you see termites, they are also present in symbiotic association with trichonympha. Okay, so these organisms will help in the digestion. So if you see wood termites, right, they go on eating the wood. They eat away the wood, but how long it takes? It takes a very long time because they have to digest the cellulose in the wood. And who helps them? It is the organisms which are living inside their body which will help them. You are asking? Then coming to chitin. Okay, so cellulose is the most abundant polysaccharide in nature, followed by chitin. So where do you see chitin? In the exoskeleton, okay, of several crustacean species, invertebrates, insects, spider. So all these organisms have chitin as their exoskeleton, a protective layer, and also in several fungi and algae. So generally, if you see oceans, how much of chitin is produced every year? Nearly 10 power 14 kgs of chitin. So look at the proportion of the oceans. How much of chitin is present in oceans? Who is giving so much of chitin there? The crustaceans living inside these oceans. They produce so much of these, and they cannot be easily digested by the vertebrates. Alright, so they form the second most abundant polysaccharide. So if you see the structure of chitin, how it differs from the structure of cellulose is that instead of glucose residues, it is a homopolymer of N-acetyl glucosamine. Right? I hope you all know what is N-acetyl glucosamine. Right? We already studied that it's an amino sugar. Right? So the homopolymer of N-acetyl glucosamine linked to one another by beta 1,4 linkage will give us chitin. Okay, exactly the same structure, that C2 is aminated and then it is acetylated. So that's how it becomes N-acetyl glucosamine. And this is the second most polysaccharide. Right, so shall we proceed?
Please go a little slower.
Slower. So why I am a little fast is because first and foremost these are basics, and so that you will get more time when it comes to certain topics which are more weightage in CSIR exams. Right, so from these topics you will generally get questions, particularly from section B. I cannot promise you it is going to be only section B. You can get section C questions, but there are not too many analytical questions from these topics. Right, so that's why I am just brushing up, because I am already having a perception that you have studied these carbohydrates already, right? So that's why. Anyways, for heteropolysaccharides, I have to be in pace with you there. So can one of you just give a brief? Homopolysaccharides. What are the homopolysaccharides?
Linear chain of same residue.
Not linear chain. They contain the same monomeric units. Okay?
Hetero is more than.
Okay, more than one. Just be open with your answers. We can correct them.
Both branched.
Both branched and unbranched. Homo, same monomeric units.
Homosaccharide forms as storage product, and heteroside.
Storage and structural, both. Alright. Homopolysaccharides. Heteropolysaccharides, they are present mainly, their role is the structural role. Alright. Their role is in the biological system. They have structural role. Not only that, they help in interactions. They help in certain binding of proteins. There are several roles. So we will try to understand the roles of these heteropolysaccharides. Right? So here are a few examples of heteropolysaccharides: glycosaminoglycans, peptidoglycans, and you have the glycoconjugates. That is the carbohydrate in association with proteins or lipids. So they can be glycoproteins, proteoglycans and glycolipids. So when I am explaining the structure of each of these, you will understand what these terminologies are. So when I say these classifications are not completely distinguished. Alright, so because if I say glycosaminoglycans are heteropolysaccharides, do not have a perception that they are only polysaccharides. Alright, along with certain carbohydrate residues, it will have some proteins, it will have some lipids in association, because their role is complex. Alright, they are all having structural role. When they are present in the structural polymer, they have to have certain receptors, right? Certain ligands on their surface. So all because of the presence of these, they interact with the proteins. So they will be associated with proteins also, lipids also. So exact classification is not, it's not bifurcated exactly the way I have shown in here. Alright, so first we'll try to understand the most simplest, the peptidoglycan or murine, about which you must have studied, right? Where do you see peptidoglycan? The cell wall of bacteria. So it is the rigid component of the bacterial cell wall and it envelops the entire bacteria. So how it helps the bacteria? It prevents the bacteria from any kind of external pressure. Alright, the osmotic lysis of bacteria is prevented. Any external pressure, it gives protection to the bacteria. Not only that, it will also help in movement of solutes or ions through the cell surface, and not only that, it confers the virulence or the pathogenicity of the bacteria, whether the bacteria is pathogenic in nature or not. Okay, so that determines by certain factors which are present on the cell wall. Sometimes even the peptidoglycan layer itself is enough to give us some infection, isn't it? It need not be the entire bacteria which we get infected. Even a small portion of peptidoglycan is enough for us to be infected. So that level of virulence also sometimes it confers. Alright, and do you see them only in gram positive or gram negative? You see peptidoglycan layer in both gram positive and gram negative bacteria. So what is the difference between gram positive and gram negative? It takes up stain. I don't want that answer. Okay, because gram positive takes up gram stain, negative does not. Tell me something different.
Gram negative is pathogenic.
Are you sure they are only pathogenic? You have both gram positive also, you have pathogenic species. That is not a typical way of differentiating.
Second layer is thicker
gram positive.
Are you sure it's gram positive? No. Peptidoglycan layer is thick in one of the species and thin in the other species. Which species?
Gram negative.
Gram negative. Anybody here? Online students?
It is thick in gram negative.
It is thick in gram negative. Thick in gram positive. If I just ask you question little confusingly, then
Gram negative, ma'am.
lipopolysaccharide. Somebody has a completely different term. Are you very sure, Praveen? Lavanjika.
So gram positive. Gram positive.
Come to a final conclusion.
Gram positive.
Gram positive?
Yes.
It is thick. Alright. The peptidoglycan layer in gram positive is thicker. Alright. And the peptidoglycan layer in gram negative is thinner. Alright. So there are several other differences. Alright. You have the peptidoglycan layer in between the outer and the inner membrane. In the case of gram negative. Alright. In the case of gram negative, it is outward, outside the membrane, inner membrane. Alright. There is no outer membrane in the case of gram positive. In the case of gram negative, it is in between the outer and the inner membrane and it is thinner than the gram positive species. You're not clear with it?
No, I'm clear.
Outer and inner membrane.
Gram negative will have outer membrane. In between the after the outer membrane on top is the thin peptidoglycan layer, on top of which it has got a thin inner membrane layer. Outer membrane, then peptidoglycan and inner membrane.
The stain is being blocked in the that concept, no? That it is getting the pores are formed. Then after
This porosity is there in gram negative. Okay, but gram positive does not have pores. Alright, this gram positive and gram negative is not our topic here. It is peptidoglycan. So we'll just come back to this. Okay, so what is the structure in gram? In the peptidoglycan, if you see, it is beta-1,4 linkage between N-acetyl glucosamine and the N-acetyl uramic acid. Alright, so I revised as soon as I started the class because I want you all to have the picture. What is N-acetyl glucosamine? What is N-acetyl uramic acid? Alright, so they are linked to one another by the beta-1,4 linkage and they form a linear polymer chain of one another, and several such linear polymer chains will lie side by side and they will be cross-linked by short peptides. Alright, so small protein sequences are present in between the linear polymer.
Ma'am, what is the difference between uramic acid and uramic acid?
Uramic acid is for mannose-amine. Mannose-amine along with pyruvic acid, which is N-acetylated, is sialic acid. That will also have some structural role when I am talking about it. Alright, this is uramic acid. Then I said there are short cross links, right? So in the case of gram-positive, you also see the presence of pentaglycine, and how these layers can be broken down. The bacteria can be killed is the lysozymes. So lysozymes are certain enzymes. Where do you find them? Where? Lysosomes. No. Tear glands. Alright, secretions of vertebrates like tears. Okay, so there you find the lysozyme enzymes. Okay, so as soon as some kind of a dust enters our eyes, the first line of defense mechanism is the release of tears, right? So they have the lysozyme enzymes. So sometimes if the foreign object is bacteria, then it gets lysed. Where at the beta-1,4 linkage between the NAM and NAG.
So lysozyme can break the peptidoglycan
Linkage between, it's an enzyme which acts on this glycosidic bond. Alright, then same way penicillin is an antibiotic, right? So that also helps in, it prevents the cross-linking. So the further formation of the cell wall does not take place. So how it looks here, you can see NAG, NAM both are linked by beta-1,4 linkage. Alright, here you see it is the tetrapeptide cross-linking. So you have a linear polymer of NAG and NAM alternating. Okay, several such layers of them are present side by side, and you have a tetrapeptide. And sometimes, particularly in the case of gram positive, you also see pentaglycine residues which are present. Alright, so here in the case of gram negative, instead of tetrapeptide, you also see pentapeptide residues present. So how this looks? So this is NAM, you have the NAG, all linked to one another by the glycosidic bond, beta-1,4 glycosidic bond. They are present as layered one next to the other, and they are cross-linked by the tetrapeptide, and also you have the pentaglycine. So this is the entire structure of peptidoglycan. Alright, so how it is present in the bacteria? So in the bacteria, they are coiled. Okay, several such peptidoglycan layers will be coiled like a right-handed helix, coiled around one another to form a tubular structure, the tube-like structure, and it goes on rotating around the entire envelope. It envelops around the entire bacterial cell. So that is how the peptidoglycan forms a thick, rigid covering over the bacterial cell and gives it the protection from the osmotic pressure or any external environment. Clear? Tetrapeptide is a cross-link. It will connect the two adjacent layers of NAM and NAG. Alright, NAM and NAG are linked as one layer. Several such linear polymers are just stacked one next to the other, and they are connected by this cross-linking between two tetrapeptides. Okay, and that will not be sufficient. So for that, a pentaglycine will be added on. That is in the case of gram positive. For gram negative, it will be a pentapeptide that will cross-link them. Alright.
Alpha coils.
Coil like
Just a right-handed helix. Alright. They will just coil and form a tube-like structure, and several such that it will become like a wire. Alright. So you can imagine a wire which is wound around the bacteria, and how this wire has got peptidoglycan layers which are helical like a right-handed helix.
for gram negative, there is no pentaglycine.
No, there is no pentaglycine for gram-negative bacteria.
So ma'am, in gram positive, tetrapeptide, pentaglycine.
Both are present.
Pentapeptide also is there?
Pentaglycine and tetrapeptide.
Okay. Gram negative?
Pentapeptide.
Only pentapeptide.
Pentapeptide. You don't see pentaglycine in the case of gram negative. You will have some exceptions, alright? When you go to find the information, you will come across variations in these from organism to organism, depending upon their virulence and all. You will have some variations. So here, apart from the peptidoglycan, you need to know certain other oligosaccharides which are present on the organisms. For example, Gram-positive bacteria has got teichoic acid on its surface. So these teichoic acids and O antigens are going to confer the virulence to the bacteria. So as I started with, I mentioned that virulence is conferred by the cell wall of the bacteria, right? So who is giving that virulence is the presence of additional groups, alright, like the teichoic acid. So if you look at teichoic acid on its backbone, it has got the ribitol or any glycerol or ribitol. Where did you hear that? Alditols, right? The reduction of monosaccharides gave you alditols, and ribitol and glycerol is one of their examples. So these ribitol residues are linked by phosphodiester linkages. Okay, they alternate with the phosphate group, and you have the OH group being replaced by NAG or alanine residues. Alright, they also alternate. So this forms the teichoic acid structure, and this teichoic acid along with the peptidoglycan will confer the virulence in case of gram-positive bacteria. In case of gram-negative bacteria, teichoic acid is absent. Instead, certain unusual monosaccharides are present called as the O antigens. So they will again confer virulence in case of gram-negative bacteria. Alright, so that's about the peptidoglycan. Next, we need to understand the glycosaminoglycans. So what are glycosaminoglycans?
So virulent are only gram-negatives.
No, both O antigen and teichoic acid, they confer virulence. These are the antigens which are determining the virulence in both gram-positive and gram-negative. In the case of gram-positive, it is teichoic acid. In the case of gram-negative, it is O antigen. So both are the virulence factors present associated with the peptidoglycan. Alright? You want it hotter? You want to reduce?
I want hot.
You want hot. Okay.
O antigen are present in gram-positive.
Gram-negative.
Gram-negative.
But even sometimes without peptidoglycan alone can render the virulence, right?
Yes.
No need of teichoic
Yeah.
acid.
These are additional virulence factors. Alright. So let's proceed to understand what are glycosaminoglycans. Alright. So the extracellular space around the cell is generally gel-like in nature, right? It is consisting of gel-like extracellular matrix, also called as ground substance. So the main composition of this extracellular matrix is the glycosaminoglycans or GAGs. Alright. So if you generally have if you're buying a bike, right, you need to fill petrol. Petrol is the source of energy. Without petrol, it cannot run. Right. So in the form of petrol, we need carbohydrates in our body. Right. We need to eat some carbohydrates so as to get energy to do our activities. Apart from that, a bike has to have screws, bolt, nuts, right, whatever, all those, and it needs suspensors, shock absorbers. It needs lubricants, right, oil. Without all these, the vehicle is going to show some or the other malfunction, right? So the same way, like how you have the uneven road, then you need suspensors. Without that, the vehicle is going to have a problem. Without oil, without lubrication, the vehicle will not have smoothness. Right? Same way, our structural component. Alright, the structural components in an organism must possess some kind of lubricants. It must possess some kind of shock absorbers. Alright, apart from the petrol, apart from the food what we consume in the form of carbohydrates will also have roles like lubricants. It will have roles like shock absorbers. Alright, several roles it plays in the body. So in the structure, the main role is to either act as a lubricant or shock absorber. So around the cell, it has got gel-like substance. So that will be acting as the shock absorber, preventing the cell from lysis. Alright, sudden change in the environment, some uneven roads, okay, some uneven circumstance will prevent it from undergoing shocks. So these are all certain factors which will help in that. So where in the cartilage, tendons, skin, blood vessels, along with certain proteins, fibrous proteins like collagen and elastin, the glycosaminoglycans are present. So these are nothing but heteropolysaccharides along with certain fibrous proteins. They form a mesh network. Alright, severe mesh-like network. Right, and they are present in the gel-like matrix of the ground substance, and they confer high viscosity or elasticity to the cell, around the cell. In the extracellular matrix, they are highly viscous and they give elasticity. So this elasticity is the shock absorber of the cell, prevent it from undergoing any kind of damage. Alright, so what are they? How do they look like? Alright, so these are linear polymers. That means there is no branching, and the repeating units are disaccharide units, not monosaccharides. So what are the disaccharides? Let's see those. One is N-acetyl glucosamide or